Well, thank you very much, Camilla, for the invitation up here. It's really wonderful to be back here in this beautiful room at Tattersalls. I'd also like to thank the sponsors for today, to Beaufort Cottage Educational Trust and to Gerald Lee Educational Trust. It's a really wonderful event to have people to be able to get back together again face to face. So it's probably quite fitting that we're in Newmarket to talk about this project, because this is really very much a collaboration between veterinary surgeons, primarily in Newmarket, but we also work up in Yorkshire, and we've got some contributors from Ireland as well, and also with stud farms. So it's it wouldn't be without all of those people that this project wouldn't have gone ahead, I don't think, because it's been a bit of a mammoth sort of long effort collating these. So what are we going to sort of face today? Well, this is something you'll see whether you are on a stud farm or a veterinary surgeon visiting those. You have a healthy pregnancy that you detected at your first scan, and essentially you scan it a week or two later and it's collapsing, it's failing, and has no embryo proper. So essentially what we've been working on for a number of years now is understanding why. And when we entered this work, about sort of 9 years ago, we really started this project. In about 80% of cases, when we went through clinical records, had no attributed underlying cause. So they were being particularly difficult to manage. So when we think about early pregnancy loss, I use this term because we've really extended the work we do up to 65 days of pregnancy. And the reason we do this is that we study both the embryonic period, so in the horse, the fetus is formed by 35 days into gestation, which I'm always constantly amazed, actually, that organogenesis, all those organs are formed like only a month into pregnancy, then essentially thereafter we've got this massive growth period in fetal life. So we think about both early embryonic loss and early fetal loss. Now, my reasoning for sort of extending to this period, not only from the fact that when we do epidemiological studies, there is a decent number that are actually lost around 40 to 65 days, but also you can imagine implantation in the horse is actually quite late. So if we get implantation failures, you would not expect these to present to sort of between your sort of 40 to 65 day scans. So what about in thoroughbreds? There's been we've heard earlier about the fantastic records that the thoroughbred industry holds, and we've certainly used these, as have some of our colleagues around the world, to try and sort of understand the incidence of pregnancy loss at various periods. And these are sort of a number of studies here, but you can see here, for example, in the UK, back in 2002, about 7.4% of pregnancies were lost before 42 days. And then we repeated this study over the 2013-14 period, and about 6.4% were lost. And if you look across studies, it sits somewhere between, say, 5 and 10% of established pregnancies are lost over this embryonic fetal period. What's quite interesting when I talk to vets or to stud farms when they're interested in data, or they might come and say, this year we've got some unexpected sort of changes in our reproductive efficiency, what we do tend to find when we look at the data is often characteristic of maybe a shift in the mare population, a reduction in maidens in those mares. And so much of the variation you see across these studies is really to do with that background mare population as opposed to necessarily management. So this group here is primarily involved, obviously, in natural mating, and this is a study done by Tom Stout over in Utrecht and his colleagues, and it's quite interesting if you look at pregnancy loss. So these are pregnancies that have been established and then subsequently lost at 45 days here. You can see with natural mating it's about 6.1%, AI 5.9%, and if you do AI embryo flush, ET, it's about 4.4%. So there's very little difference actually here in pregnancy loss rates with these techniques. But when you move over to ovum pickup here and ICSI, an embryo transfer, you can see much higher rates of embryonic loss. So I think this is quite interesting, and we're dealing with probably relatively similar mechanisms here when we look at a population as a whole. So we've got this challenge to sort of, but one of the things that we were really interested in is trying to understand the underlying causes, because sort of going into this initially, a lot of the approaches were really quite generic, because if you don't know the cause in 80%, you've really only got options to you at looking at a couple of treatments. which you're not really sure whether they're targeted. So that's where we focused our attention. And what I'm going to present you to today is some data which would suggest actually it's genetic variants, so genetic changes within that early embryo that are probably going to be the most common cause of early pregnancy loss. And my best estimate at the moment is they're probably going to be representing about 50% of these cases that you see. Endometrial disease, we've heard some beautiful talks by Karen Wolfsdorf earlier about the barren mare and endometritis. And when we've looked at the association with infection with early pregnancy loss in our studies and other studies, it tends not to be over more than sort of 10% of early pregnancy losses that will have some sort of association with endometritis. So it's clearly important, but not the bulk of them. The other condition you could think about, we know that endometrial cysts are associated with increased risk of pregnancy loss. That could be due to the cyst, and some studies have shown that, but actually it could be just a representation of underlying changes in the endometrium. We heard something about fibrosis earlier, so these sorts of changes will increase a mare's risk for pregnancy loss, and I would estimate these are probably around the 10 to 15%. And then we've got a mix of what I call other, which is about 25% of the others, and this is a real mix where there'll be relatively small contributions from lots of different breakdowns in mechanism, and we're still trying to understand those. But as you can imagine, depending on this underlying cause, the treatments and approaches will need to be quite different. So here is some data. I just touched on this then, but this is looking at a population of mares that experienced early pregnancy loss, and you can see here about sort of 18 to 20% of that population, those mares were found to have uterine cysts. These won't always be causative for that pregnancy loss, but it gives you a sense of the proportion of the population. If you look over here, though, when we've accounted for things like mare age and status and various other confounding factors, the odds ratio of a mare suffering pregnancy loss if she has an endometrial cyst is about 1.7. So if they don't have cysts, our EPL rates are around 6.6 and 14% if they did. But one thing that jumped out here was when we did this work is actually, you can look down the bottom here, we found about 83% of these that we studied had a negative cytology and culture. Some of these could be false negatives, and there are other ways to investigate that we've heard about today, but a good portion probably aren't. And also when you look through clinical records, about 75% didn't have any notes of uterine pathologies. So clearly there were some other sort of causes that we needed to think about. And that's how we got onto genetics. So I'd just like to pause here and say Charlotte Shilton, who's a PhD student in the laboratory, has really led all this work that I'm going to present today and done all this genetic analysis. So she's just finished, for those that know her, and successfully defended her viva, which is fantastic. So when we think about genetics of what? And I think this is a really important distinguishing factor because both of these will be important. So if we think about the genetics of the mare itself, she's essentially the environment in which that embryo is being nurtured and developing. So we could think about genetic variants in that mare that might affect her endometrial environment, her hormone production, and her immune responses during pregnancy, which we know must be modulated to the conceptus. But on the other, what we tend to find in the literature is these tend to modify the risk. So these sort of variants that have been described modify the chance of a pregnancy loss. On the pregnancy side, the genetics of the fetus and the placenta, remembering the placenta matches that of the fetus, we can find what we call lethal variants. So these are genetic changes that resulting pregnancy possesses that we think are incompatible with life. And so we know that they are not found any live-born or any adult individual, and that's what we call a lethal variant. And they could come either inherited through the germline, so they could come from the egg and the sperm, or they could be acquired in early development. So they could be during that really rapid phase when that embryo is developing. Now, all of these things also are going to be interacting with other external factors that can impact on these events. So there are a number of, when I've talked about genetics more broadly, I'm now going to talk about it in a little bit more detail. So what do I mean by that? Well, this is from a review article, and this is the size of the variant on the bottom. So the code of the genome, you can have as small as one base pair change all the way out to changes in a whole chromosome. And so we've been interested, and we started here on what we call trisomies or monosomies. So these are aneuploidies, where you get a whole duplication or loss of a chromosome. You can get copy number variants involving over 1,000 base pairs of a chromosome. You can go all the way down to 1. So we're sort of moving our way along this line to understand the various contributions of each of these. What you might be more familiar with are these structural autosomal translocations, and I'll talk about those in a second. So I've got those different types of variants, and we've talked about the 2 compartments. So I'm now going to have a little think about that MARE first. So what sort of genetic invariants inherent to the MARE could contribute to early pregnancy loss? And this is the best one that's described. It's been described for many years now, and these are what we call structural translocations. So essentially, you can see here that part of a chromosome here has translocated here to another spot here. And these are MAREs that present with recurrent pregnancy loss, and these can be detected with karyotyping, and I'm sure many people here have done this before. But what's really interesting here to note is these are relatively uncommon, so they would account for about 2% of early pregnancy losses. Important, can be diagnosed, but at relatively small contribution, and probably less than 0.2% of mares, breeding mares. They can be diagnosed easily with the blood, and we send them over to Texas A&M at the moment, to Terry Rodsip, who we've heard about a little bit earlier, with blood, and they can produce a live foal, but they have normally a history of multiple pregnancy losses. The other sort of genetic variants that we don't know much about in the MARE, so I'm just going to point to what we might look for in the future using this data from human, is SNPs that are found in the MARE that might change her function. And we did this, had a student who did this actually in COVID when we had to quickly switch to an online project, and she went through the literature and she found there are actually 164 publications involving single base pair changes in women that were associated with changes in 81 genes that were increasing the risk of pregnancy loss. And we haven't actually looked at this in any detail in the MARE, but there is one study actually in thoroughbred mares that found a SNP in a gene called P53 that was associated with an increased risk of abortion. So this is a reminder, this is a change in risk. This is not necessarily lethal. So now we're going to move into the fetal compartment. In this compartment, we're really interested in the lethal variant, so what will cause those pregnancies to fail. And probably the most common thing described in women is aneuploidy, where you get a loss or a gain of a whole chromosome. And you can see here, this is an example here of a trisomy 1. So you can see the 31 autosomes here in the horse, and there's 3 copies you can see here. Now, when we screened all the early pregnancy loss samples that had been submitted by clinicians here in the UK and in Ireland for this condition, we found 16% of them had 3 copies of a chromosome and 5% had lost a copy, so only had 1 copy. And we believe that, with the exception of one case here, that these are all lethal because they'd never been described. So establishing this now as what appears to be a common cause of early pregnancy loss, you can see here its prevalence. It's occurring in 20% of the early pregnancy losses that we've screened. And I'll show you the gestational age. These are biased probably towards those that are over sort of 20 days, although about 18 of these were under 30, I think. So we've got a reasonable number in there that are earlier, but there's a bias towards 42-day scans. We also extended this because we wondered, well, if we're finding this in early pregnancy, if you look down the bottom here in adults, we don't find these aneuploidies in adults. There are actually 12 live-born horses that have been described across the literature that have aneuploidies that are compatible with life, so 12 only. But we're interested in the intermediary phenotypes, and we identified one case of abortion here with a partial trisomy and a case of stillborn. So it seems it's primarily a loss in early pregnancy, but can contribute to abortion as well. So it looks something a bit like this now, where you've got your early pregnancy loss 20%, we drop right down here to sort of 1 to 4%, and extremely rare cases here in adults. But what I'd also like to mention is actually it's likely to be contributing to these preclinical early pregnancy losses. So these are these mares that have probably conceived, but they're losing their pregnancies prior to the 15-day scan. So these are the gestational age of the population that comes into us here. So you can see here it has got a median age of about 42 days of gestation when they're submitted, and they range here from about 16 days. We've got one very late one up here, but we see these trisomies and monosomies across this age. So you can see on the right here, these are the MARE ages. So this is not a condition just of old mares. You might be thinking that maybe it's going to be biased. And we think it probably with time, when we can look at more of these, it may well be. But we were able to see these aneuploidies in middle age and also as young as 3. And actually, when we went back into the literature, teenage women that conceive and lose pregnancies have much higher rates of aneuploidy as well as older women. That is better known. And here's the gestational ages of the early study here. So what about, you're probably thinking, well, was this good or bad? What happens if she conceives an aneuploid pregnancy? What's interesting, these are really small data to start with, but in a small population of mares, they lost their pregnancy early enough that we could look at whether they were covered again that season and whether they conceived. And if you look here at the aneuploid EPLs, actually there was 4 mares that had these aneuploid in time to be recovered, and actually 3 of them ended the season with a live foal. So it's not necessarily a bad thing that they conceive these aneuploid pregnancies, but it would be important to differentiate them from this group here, because these ones here, which weren't aneuploid, they had a very poor percentage of pregnancies later. So these ones are going to be difficult to work with. What do they look like? So they had a range of sort of challenges, obviously. This one here had no, this is what we call anembryonic, so there was no embryo that developed in this when we lose a chromosome here. This is chromosome 31, and this one here has lost chromosome 27, and you can see here it's really swollen and edematous and has some sort of vascular disorder and looks markedly different to this normal fetus here. So where do these come from? That's, I guess, probably what you're going to ask me next. Well, actually, what we think, our hypothesis at the moment, is that the majority of these, not all, but are likely to be coming through a defect in the egg, so during meiosis. And so this is some work done by Marta de Ruta Villani, who's over in Utrecht, and she's looked at aneuploidy in eggs from mares that are young and old. So these are 14 and under and 16 and over. And you can see the amount of aneuploidy in these eggs is substantially higher in these older mares. The other thing that made us think that these are coming through what we call through the germline, so they've been inherited from either the mare or the stallion, is that when we tested different parts like the fetus and the placenta in multiple samples, we always found these genetic changes, suggesting that it had actually come through this, what we call through the germline. Although we can't clearly rule out, and there was likely to be some contribution where this has been introduced in early development, and that's what we see in women. So whilst we were in a whole chromosome there, we were also interested in the next level of variant. So we're going to move up that line that I showed you. So the next variant we're interested in is something called copy number variation. So in these cases, here's our chromosomes here. And if you look here, this is this segment here, C, and in the normal here, you've got 2 copies. If you see here, this C segment has been lost. And this is what we call a deletion, a microdeletion. And on this side here, we've got a duplication of this. So this is a microduplication. So a segment of the chromosome has been duplicated. So when we looked at our pregnancy loss, we've excluded now all the aneuploidies. So we're going, okay, what's left in this group? And we actually found that these were highly enriched for these copy number variants. So the genome, up to 3% in this particular case here, of the genome was disrupted by this copy number variation. So this type of disruption, this level of disruption, is probably very similar to a trisomy that I just showed you of a whole chromosome. So we believe that this is likely to be contributing to that pregnancy failing. It occurred in 9% of these pregnancies that we screened, so it wasn't a small number as well. And the underlying mechanisms are probably not dissimilar to what we see with aneuploidy. There have been similarities as far as the mechanisms that lead to these 2 events. But as well as this sort of widespread effect of copy number variation across the whole genome, and it's all been sort of disrupted and disrupting the expression of genes, which then in turn disrupts development, We also found that if you get deletion of a specific region that carries a key gene in development, this was also associated with that pregnancy failing. So this is an example here of a deletion we found on chromosome 20. And this included a tubulin gene, and tubulin is a gene that's really important in development of the central nervous system. And so it was including a gene we think that fitted well with it being lethal, and we know that when we've looked at these embryos before, we often find disorders of the central nervous system. The other thing that was interesting is they seem to all be lost at a relatively similar time, between 35 and 40 days. And this is an example of one of the fetuses from a pregnancy carrying this deletion that had a neural tube defect. So essentially the neural tube closes during early development, and this has failed to occur. And this has been associated with pregnancy loss, quite well described in women. So now we've got our big pieces, our sort of intermediary pieces of the chromosome. So the next thing we're thinking about is SNPs, so going down to those smallest regions. But before we did that, we wanted to ask a more general question around the effects of inbreeding on early pregnancy loss. So why were we interested? Well, inbreeding is going to increase the homozygosity of the genome, which we know in turn can decrease the fitness of the population. So if you've got two copies of the same, they're more likely to contain sort of a lethal variant because they match essentially. So what we were measuring here are what we call runs of homozygosity. So runs of the genome which are matching, inherited from the maternal and paternal side. And we've used here a measure called the inbreeding coefficient to do this work using genetic sequences. And what we found is actually that these EPLs were less, if anything, they've clustered into two groups, but the level of inbreeding actually wasn't increased in the EPLs as we hypothesized it could be. You can see here it's very similar levels to adults. But we know inbreeding does affect fertility in other areas. It reduces semen quality, can be associated with retained placenta, viability of foals, and there's a little bit of conflicting data in the literature around fertility, which is interesting, so maybe that fits with the EPL. So don't rush away and think that inbreeding is a good thing, but what we know in the context of early pregnancy loss specifically, that it is not going to be a major contributor. But what we also know is that this is looking more generally at the genome, and it tells us actually it's probably more important for us to look at specific SNPs, single nucleotide polymorphisms, that are important at leading to pregnancy loss. So we can't take a general approach saying, regarding inbreeding when it comes to this particular outcome, it is about thinking about specific SNPs. So we're up here now. So what SNPs do we know that are lethal, that are inherited and lethal in fetal life? Well, actually, in early pregnancy, there are none identified to date. And this is our next question to tackle. There is one that you might have come across that's been reported in the literature. And what they did, this is a gene here, an NK cell gene, which is an immune related gene, and this particular haplotype was found to be absent in live horses, so they speculated it might be responsible for early pregnancy loss. We've actually tested all our bank and haven't been able to associate this particular SNP with early pregnancy loss. So it could still be maybe before 15 days or during other periods. We can't rule that out. Probably the SNP that's best known, not for early pregnancy, but for abortion, is a SNP in the PLOD1 gene that was originally described in warm bloods and more recently in a thoroughbred, which is associated with lethality as well. We know this, though, is not associated with EPL. So just a summary of these lethal genetic variants that we've found on the fetal placental side. So if we have an aneuploid genetic background, we know that can result in primarily early pregnancy loss, but also can abortion. That this was 20% of early pregnancy loss cases we screened. We know there is an example with just under 10% where widespread copy number variation is going to disrupt development. And we've got examples here of specific CNVs, so a deletion involving a key gene as well. And then obviously when we look later on, homozygosity for this SNP in PLOD1 is associated with abortion. So the next thing to do is, I think, to also go back and think about the MARE on the MARE side. So looking forward, where are we heading with this? So we are thrilled to have some funding actually from both the Cottage Educational Trust and also from Gerald Lee to sort of take this work forward, thinking about diagnosis of these genetic variants. So can you predict whether a MARE is carrying them? There's two ways to approach this. The first is you could actually submit the abortion material or the EPL material to the laboratory. And at the moment, this is just done in our laboratory by clinicians sending in samples. We isolate the DNA and we determine whether there's an aneuploid present. So that's a one viable way, but that's always retrospective diagnosis. But what would be nice is to be able to predict whether a MARE is carrying a fetus with one of these variants. And to that, we've actually turned on a methodology that's used and now well established in human medicine. And that is here, you can see the fetus with the placenta here. We know the fetal DNA that's coming out primarily from the placenta here, it's being excreted into the maternal bloodstream. So you can actually take a blood sample here of maternal blood and isolate this fetal DNA, and then you can look for these variants in that. So that's what we're looking at developing at the moment, and we've just bled our first mares to and isolated some of this DNA. And so we'll look forward to updating on that over the next year or so. The other thing to keep in mind in regards to these variants is you can test the mare and stallion. So if it's something like PLOD1, that's a very predictable condition. By testing a hair sample in a mare and a stallion, you can predict whether if they're mated, it would have a 25% chance of a loss. So what about what can you take away from these genetic variants? They could probably be quite frustrating because if you're carrying a lethal variant, you know that pregnancy is not going to go forward. But I think they can really fit into the management of these cases. The first thing is actually by knowing the underlying cause, you can then reduce treatments such as that might not be necessary, progesterone, antimicrobials, other things that are targeting the endometrium, but when you've got a genetic variant, they're not going to necessarily help. By doing that, by understanding the cause, we can also improve antimicrobial stewardship, which we touched on earlier. We can also ensure pregnancies that are carrying a lethal variant are not prolonged, so you don't want to be treating those pregnancies if they have a lethal variant in an ongoing manner. They would be best to fail and those mares to be rebred. And as I just mentioned, you can use some of these variants to use it as a guide of it's a bit more like a combination of compatibility. So if we can identify which SNPs are lethal, homozygously lethal, we can then actually guide your mating decisions and you can avoid, rather than at the moment, I think you probably breed to a stallion, she loses a couple of pregnancies and then you move to another stallion. This way we could actually predict whether that would happen. So I'm going to touch at the very end as I finish up now about looking forward to the future as well as far as treatments. And there's some interesting data coming out. This is very much in mice at the moment, but so don't rush out and start feeding your mares this vitamin. But I wanted to really highlight this of the types of things that might come through in future years for things such as aneuploidy. So niacin is a water-soluble vitamin B3, and its form in food is it's in nicotinic acid and nicotinamine, and also we know tryptophan can convert into nicotinamine within the body. And what they did in this study here in mice is they fed this nicotinamine in the drinking water of maternally aged mice, and they looked at oocyte quality and embryo development. And if you look here on the right, these blue ones here are the young, these are the aged mice here, and these are the aged mice had this nicotinamine supplement, and the rates of aneuploidy have dropped. Now, there is clearly much to do on this. This is very preliminary. These are in experimental animals. We have no idea if this is going to have an effect in mare. But I put this up as a proof of principle that actually something like aneuploidy and these genetic variants, particularly if they're originating in maternal meiosis, which we think they are, may have some targets in the future for us to reduce those occurrences. And indeed, a paper's just come out last month out of Australia where they have been giving this nicotinic acid to mares, and then they've been measuring it in the follicular fluid that bathes the egg. And what they found is that it is able to be taken up in relatively high concentrations in those eggs. So There might be something in the future, but this is very much experimental, so just put it in that, I'll leave it in that context. So hopefully now I'm going to sort of round back to where we started. This is going to give you some ideas of the underlying causes for the pregnancies, whether it be genetics as the first thing to think of, then the endometrial environment, and then a large number of other smaller contributors next time you look at the MAREs. So just a quick acknowledgement of all the people, very importantly, this is very much a collaboration. We work closely with Terry Rodsop and Brian Davis, geneticists at Texas A&M, and Charlotte and Anna were the main contributors to this work, along with Jess Roach, who's been working on abortion. And I'd also like to thank all our funders and our collaborators, clinical collaborators that make this possible. So thank you very much for your attention and again for having me here today.